Hello, you're welcome to our lectures on biological techniques. Our lecture will be on the collection, preservation, and identification of zooplanktons. Now, what are zooplanktons? Zooplanktons are minute animal or animal-like creatures that are seen floating on the water body. And because of their minute nature, they are unable to swim against the water current. So they float on the water surface and they drift along with the water. They drift along with the water. And they include organisms such as cnidarians, minute cnidarians, and other organisms like the copepods and others. Even the larva and pupa of some benthic organisms. The larva of some benthic organisms usually were planktons, and then when they become adults, they settle at the bottom. So they there are temporary planktons and we have the permanent planktons, temporary zooplanktons and permanent zooplanktons. The permanent zooplanktons are the ones that do not change into any other form to become benthic organisms or pnictons, but they are permanently on the surface, while temporary planktons are the ones that usually are found on the surface of the water body at some stage in their life cycle, particularly as larvae, like the larvae of the crustaceans are usually temporary planktons. The larvae of benthic organisms, which also include crustaceans, mollusks, and others, are also temporary planktons. They float on the surface, and then as larvae, when they are minute, and then when they become adults, they settle to the bottom. So we are actually looking at the procedure for the collection, preservation, and identification of zooplanktons. The collection, preservation, and identification of zooplanktons. To collect zooplanktons and preserve them, we need 10%, 4% formalin and distilled water. And the apparatus include a graded fleet of nets, that's plankton nets, usually 100, 150, and 200 micrometers mesh size. Then a Sedgwick rafter counting chamber. This is the plankton net. Okay, it has a frame, a rope, and then a net, as well as a bottle at the bottom. The Sedgwick rafter counting chamber is used in place of the microscope slide to identify the planktons. Then you need a drop-in pipette, then a microscope, and a 50 ml glass vial. You need 50 ml glass vials. Now I want to look at the procedure. What's the procedure for the collection, preservation, and identification of planktons? The procedure includes the following. First, you filter a known volume of water through the graded fleet of plankton net. Okay, a known volume of water is filtered through the graded fleet of plankton net. And the filtering can be effected by towing the plankton net vertically or horizontally over a specific distance, m, or over a specific time. So the net is towed vertically or horizontally, vertically or horizontally. Now, this is the procedure. First, you drop the plankton net into the water while the boat is not moving. And then when you drop the net, you now move the boat, you propel the boat. And as the boat is moving, the net is used to collect surface water. Okay, you tow the net over a specific distance and over a specific time. Then after towing the net for a specific distance and time, the net is now raised up and the planktons are then collected from the bottle. The collected samples are now washed. We now wash the filtered samples into a 50 ml vial and preserve in 4% formalin. It's now the water, the sample is now washed into a 50 ml vial and preserved in 4% formalin. In the laboratory, you now make up the volume of each vial to 200 mils using distilled water. Okay, you make up the volume of each vial from 50 mils to 200 mils using distilled water. Then you now homogenize the sample by shaking properly. You now shake it properly, you homogenize it, mix it properly by shaking. Then quickly take a subsample, which is usually 0.5 mils, using a pipette and place it on a Sedgwick rafter counting chamber. Okay, a subsample is then collected with a drop-in pipette and placed on the Sedgwick rafter counting chamber. Then under a microscope at low power, this time is at low power. It's unlike the phytoplanktons that we use times 100 objective. But in the zooplanktons, we make use of times 10 objective of the microscope. Okay, so under a microscope at low power, identify and enumerate the zooplanktons using appropriate identification keys. So we use appropriate identification keys to identify the zooplanktons. Thanks a lot for watching. We appreciate. Please subscribe to our YouTube channel for more educative videos.